Hello, everyone, and welcome to Dana-Farber's Department of Data Science Frontiers in Biostatistics webinar. My name is Erica Fike, and I'm the Program Manager for the department. Before I turn it over to our organizers and speaker for today, I'd like to point out a few items. The webinar today will be recorded and will be available on our YouTube channel within the next week. You can find our channel at youtube.com/DFCI Data Science. For this webinar, the chat function has been disabled. If you have a question for the speaker or the organizers, please use the Q&A tab. Our faculty organizers will manage the Q&A session after the talk. With that, I will turn it over to Nabiha Tayob.
Thank you, Erica. Thank you everyone for being here. Welcome to the first Frontiers in Biostatistics seminar series of 2021. We're happy to have Dr. Jing Ning presenting today. Dr. Ning is an associate professor in the Department of Biostatistics at the University of Texas M.D. Anderson Cancer Center. Her research interests include the design and analysis of clinical trials, joint modeling of survival endpoints, joint modeling of longitudinal measurements and recurrent events, and non-parametric and semi-parametric statistical methods. In 2018, she was an Andrew Sabine Family Fellow, and she is the PI of an NCI-funded project, Comparative Effectiveness of Cancer Research, Use of Data from Multiple Sources, and the co-core leader of the MD Anderson Ovarian Spore Biostatistics Core. Today, Dr. Ning will present a seminar entitled Statistical Modeling for the Adjustment of Sampling Biases. Please join me in welcoming her virtually to our department.
Thanks so much, Nabiha, for the kind introduction. Good afternoon, everyone. I'm honored to have this opportunity to share our recent work on how to correct sampling biases using statistical tools. So for sure, the topic is too broad for a 1-hour talk. So my plan is to use 2 or 3 examples, depending on time, to illustrate how statistical methods can be used to obtain valid inference given biased data. So the 3 examples are survivorship bias in survival analysis, outcome dependent sampling bias in longitudinal data analysis, and the publication bias in meta-analysis. So for each example, I'm going to introduce motivations and statistical method, but I'm not going to talk about too many technical details, such as large sample properties. I will list some key references. So if you are interested, you can check the technical details in those papers. Let's start out by looking at the journal definition. So bias sampling is encountered when subjects in the target population are not given equal chances to be selected into a study cohort. The major challenge as recognized by many researchers is that the distributions of the observed data is not the same as that of the target population. Of course, it will cause problems in our data analysis because any statistics computed from the observed data has the potential to be systematically inconsistent. The bias can lead to over or under representations of corresponding parameters in the target population. So then you may ask, how can we know if the sample is biased? In applications before analyzing the observed data, we needed to think very carefully about the study designs and the sampling methods for data collection. For example, it is well known that survival data from prevalent cohorts are left truncated. So when analyzing such data, we needed to handle the survivorship bias in which only surviving subjects can be recruited into a study. Or sometimes we can identify the sampling bias by comparing characteristics in our sample to what we know about the target population. So think about the characteristics that might have important relationships with the response variable. For example, assume that gender is associated with the response variable. If the observed data include only 20% males, which is quite different from the gender distribution of the target population, then the observed data are likely to be biased. We needed to revisit the sampling method and correct the induced bias using statistical tools. So now let's move on to the first example in my talk, a prevalent cohort of breast cancer. So when studying the natural history of a disease and assessing the intervention effects on survival at the population level, the incident and the prevalent samplings are commonly used for recruiting cohort subjects. Assume we want to evaluate the survival duration for breast cancer subjects. If we use the incident sampling, we will sample a group of asymptomatic healthy but at high risk subjects into the follow-up study. We will observe their occurrences of breast cancer and follow them until their death, lost to follow-up, or end of study. On the other hand, if we use a prevalent sampling, we will draw samples from patients with breast cancer. So then these identified subjects are followed until their death. Obviously, the incident cohort often takes a long duration to observe enough incident cases for rare disease. When the failure duration are quite long, this design takes a long follow-up to observe enough failure events as well. On the other hand, the prevalent sampling design draws samples from a disease-prevalent population and is consequently more efficient in terms of time and economic consideration. However, there's no free lunch. We must be aware that the collected data from the prevalent sampling are subject to left truncation. So the subject with the initial event, such as breast cancer onset, have to survive up to the enrollment time to be included into study. So the observed survival durations tend to be longer than those in the target population, which is termed as a survivorship bias. So our motivating study for the survivorship bias is a prevalent cohort of breast cancer recurrence. So despite treatment advances in breast cancer, some patients still have breast cancer recurrence. The management of relapsed breast cancer is complex, and it is essential to evaluate the survival distributions and the association between patient characteristics and the survival after cancer recurrence. Since here the residual survival time after the cancer recurrence is the key, our collaborator extracted a prevalent cohort for targeted patients who experience breast cancer recurrences from the CMED care database, which was used as a preliminary data analysis for their grant application. So when receiving the data through the communication with our collaborators, we know that the subject with breast cancer recurrence have to survive up to the sampling time to be included study cohort in their data extraction. For example, subject 4 died before the sampling time and was excluded from the cohort, although this subject had the cancer recurrence. So our first result was just to use a standard method for left truncated data to analyze this cohort. However, after carefully checking the truncation probability, we realized that the data structure is different from that of standard left truncated data. So in this cohort, the truncation probability is determined by the overall survival time and the truncation time. However, the overall survival time is not the outcome of interest. The time measured from the cancer recurrence is a primary outcome in our particular analysis. Of course, the residual survival time is positively associated with the overall survival time. So in other words, our primary and outcome is correlated with the truncation probability, although it does not directly determine the truncation probability. So to handle such a unique data structure, we need to model the bivariate survival outcomes. So before introducing the joint model for bivariate survival times, let's look at notation first. Basically, here we use two sets of notations, one for the target population and the other for the observed data. So for the target population, to the U and to the V denote the time from breast cancer onset to breast cancer recurrence and the time from breast cancer recurrence to death, respectively. And to the A denotes the time from breast cancer onset to study sampling time. So here we can observe the bivariate time only if the sampling constraint is satisfied. So for the observed data, let UVA denote the observed biased counterparts of those in the target population. Here we assume two conditional independence assumption, one for the truncation time, the other for the censoring time. So our joint model includes several components. First, two Cox proportion hazards model assumed for the margin distributions of the two gap times. Second, a coupler model is applied for the dependence structure, which is commonly used for bivariate survival time. We are going to use a graphic method to select appropriate coupler in our data application. So the last one is a parametric model for the truncation time. So under this model specifications and two conditional independence assumption, we can derive the likelihood function of observed data. Notice that the joint density function in the double integrals involve two non-parametric components, the two baseline hazard function. Hence, directly maximizing this observed likelihood is computational prohibitive. Also, the double integrals make it very challenging to implement the profile likelihood idea, although it is successfully used in survival analysis under Cox model. So to tackle this issue, we propose estimating procedure that use the pseudo likelihood together with the two-stage EM algorithm. So specifically, we used the conditional likelihood of observed truncation time to estimate the parameter in the truncation time distribution. So you may ask, why not use the full likelihood to estimate all parameters simultaneously? Actually, our simulation study suggests that the separate estimation perform more stably compared with the MLE in our setting, resulting in better finite sample performance. So then in step 2, we use the EM algorithm to maximize the pseudo likelihood after plugging in the estimated theta from step 1. So in the M step, it is still computational non-trivial to maximize the expected log pseudo likelihood due to the non-parametric components. Accordingly, our strategy is to use a two-stage method. In the first stage, we estimate margin distributions to Cox proportion hazards model. And in the second stage, we estimate the association parameter by fixing the margins from stage 1. So then the updating parameters of a margin distribution can be easily implemented by the existing program for the Cox model with a weight option. So you can see, although the estimation procedure involves multiple steps, the computational code are not that complicated due to the closed form in the E step and existing argument for the M step. We have established the large sample properties of our estimate. I will skip details here. So if you are interested, you can find the technical details in our paper. Let's look at some simulation results on a simple setting. So we evaluate the performance of a proposed method and compare it with that of a method ignoring sampling bias. Here we call it as a naive method. For this particular simulation setting, we generate the average survival time from Clayton copula. We consider it two covariates. One is a binary and the other is continuous. The truncation time was generated from exponential distribution. In the observed data, we only keep subjects satisfying the sampling constraint. So we can see the proposed method achieved good accuracy with small empirical biases under this setting. On the other hand, the naive method yield biased estimators. It tended to underestimate the association parameter and overestimate the regression coefficients. Here, the estimated regression coefficients by the naive method had relatively small empirical biases because the truncation time in this setting was independent of two covariates. More importantly, the naive method consistently underestimated the cumulative baseline hazard function. In other words, it overestimated the baseline survival function. For example, the empirical biases were around 40% in both scenarios. We also conduct a simulation study to confirm that the diagnostic method could help the selection of appropriate copula model, even given the biased data. So this figure illustrates two scenarios, one for the Clayton copula, the other for the positive stable copula. The solid lines represent the true cross ratio curves. As expected, the diagnostic method could not accurately estimate the cross ratio curves given the biased data, but it could adequately detect the association pattern, so from which we could select appropriate model from the copula family. So now let's go back to the aforementioned breast cancer recurrence data. So here I just highlight the main messages from our data analysis. The two Cox proportion hazards model were assumed for the two gap time respectively. The exploratory plot suggests that the estimated cross ratio stayed constant over time. We then assume a Clayton copula model for the dependent structure. The Weber distribution was used for truncation time distribution. So here we can see for the residual survival time, the proposed method indicated that a higher AJCC stage was significantly associated with a high risk of death, while the naive method suggests that such an effect was not significant. Interestingly, the estimated association parameter by the proposed method suggests that a positive correlation between two gap times as expected. But naive method, the estimated association parameter indicate a negative association. So we have conducted additional simulation study to evaluate this finding. So when the strength of association is moderate, if we ignore the bias sampling issue, the naive method can produce the opposite direction in terms of association parameters in our simulation studies. So we then plot the survival functions for patients with different profiles using both the proposed method and the naive method. So the substantial difference between the difference in the estimated survival medians indicate that ignoring the sampling bias could lead to severe overestimation for the residual survival time. So now let's move on to the second example, outcome dependent.
Hi. Can I ask a question? This is Hajime.
Yeah,
So you use the copula, right, to describe the dependent structure.
yes.
And another way might be using the probability model, random probability. So is there any clear advantage of using copula against using probability stuff?
Yeah, thanks for raising this important question. Yes, I forgot to mention that one advantage to use copula is that the model and the estimation of the marginal distribution and the dependent structure is independent. So we can separate model the marginal distribution and the dependent structure. Then the misspecification of the dependent structure would have a minimal effect on the estimation of the marginal distribution. This is one advantage to use the copula model.
I see. Thank you very much.
So now I'm going to move on the second example, outcome-dependent sampling bias in longitudinal data analysis. In long-term follow-up studies, longitudinal data are often collect until a terminal event occurs. In analysis of such data, two assumptions are commonly assumed. One is independence between the observational times and the outcome. The other is independence between the longitudinal outcomes and the terminal event. In some applications, subjects often selectively miss their pre-scheduled or pre-specified assessments based on their expected outcomes. As a result, the frequency and the timing of the assessments may be correlated with the longitudinal outcomes. Hence, the standard approaches such as GE may lead to biased estimation and inference. Let's look at a typical example with sampling bias for longitudinal data. So as we know, over the last few decades, the increasing incidence of obesity has become a worldwide health issue. So among available weight control preventions, web-based programs such as Spark People have gained popularity because of their flexibility and low cost. So the main feature of these programs include education content, self-monitoring tools for daily diet and exercise, and social support tools such as blogs and friend relationships similar to other online social networks. So members use a program in a self-directed and self-paced manner. Members can selectively report their weights at each of their logins. So for the evaluation of such programs, most of analysis, including our early work, were based on the weight difference. For example, the difference in a participant's weight when entering the study compared to that when leaving the study. Inevitably, ignoring the information of weight in the middle of the study will result in misleading interpretations. For example, participants may experience a rebounder in a weight loss Participants may lose 10 pounds in the first 3 months, and then gain 8 pounds back in the next 2 months. So examination of the overall weight difference is not able to reveal such patterns. It is more informative to use weight data over time and study the effects of web-based programs during the entire study period. So then our next question is whether we can apply standard methods such as GE for this particular study. Unfortunately, the answer is no. We did some exploratory analysis and found that there was a strong positive association between favorable weight outcomes and high frequencies of weight entries. It is not surprising because a participant who is making better progress in losing or controlling weight may be more willing to report their weights. So this outcome-dependent self-report mechanism could cause the web-based weight loss program to appear more successfully than it really is. So in statistical literature, the most commonly used model to handle the outcome-dependent observational process is a joint model. So for this application, the joint model would have 2 components. One is mixed effect model for the longitudinal weight outcomes. And the other is a freight model for the intensity function of the self-reported process. So then the key assumption is that the 2 processes are independent with each other, giving the random effects and covariance. So under the conditional independence assumption, we can easily derive the likelihood function, which involves integrals with respect to random effects. So one limitation here is that we have to giving model assumptions on the self-reported process. So in our motivating example, it's not that easy to specify a reasonable model for the self-reported process. Apparently, a vector of random effects instead of a single random effects is needed to capture the data structure. So then the estimation procedure is highly computational intensive and unstable due to the multiple integrals in the likelihood function. There is a high chance to receive the non-convergence message from the software. So an alternative method in the literature is modified GE. So the basic idea is to weight the observations to create a pseudo population in which the self-reported process is no longer associated with the outcome process. So here we can see we still need to model the self-reported process to estimate the weight function as well. So if we are not interested in that process, it is not desirable to take extra efforts to estimate the nuisance parameters with additional model assumptions. So these limitations motivate us to work on new methods in which minimal model assumptions are needed. So now let's go back and look at the data structure again. So this is a density function of observed data. While the covariance effects on the response variable in the target population are the parameters of interest. Unless the self-reported process is independent of the outcome, giving conditional or giving covariance information, we cannot ignore the sampling biases to use the likelihood function of observed data to estimate the parameters of interest beta. So alternatively, we try to identify a conditional probability that is free of nuisance parameters. Actually, we found that the probability of observing a pair of observations from two subjects condition on their order statistics only depends on the density function of the target population. So it is free of the self-reported process. So by using this conditional probability, we can construct a pairwise likelihood by multiplying all such probabilities from the observed data together. So this likelihood belongs to the family of composite likelihood. It can be shown that the score equation is unbiased estimating equation for beta. The variance formula can be derived using statistical theorem. So you can find the details in our early paper. However, it is not done yet. For the self-reported data, we have additional data complexity due to the violation of the independent censoring assumption. So our exploratory analysis suggests that the length of participation in the program was strongly associated with weight loss as well. So to deal with this informative censoring, we have to use a joint model framework. But different from the parametric joint model, our proposed one is a semi-parametric joint model without specifying the distribution for the random effects and the self-reported process. So here, for the longitudinal outcome, we use a semi-parametric proportion likelihood ratio model and included a subject-specific random effects in the baseline density function. Here, the semi-parametric proportion likelihood ratio model is a generalization of the exponential family while leaving some parts of the distribution unspecified. With the informative censoring and the self-reported process, we calculated the density function of the observed data again after considering the potentially informative censoring. So seen from this equation, if we use the full likelihood of the observed data to estimate the parameters of interest, we need a correct specification of the distribution of self-reported process, informative censoring, and random effects. Of course, these specifications are all subject to potential model misspecifications, which may need to bias inference. It's better to have a more robust method. For our proposed method, we assumed two assumptions. The first one is standard in the literature of longitudinal data analysis under random effect model. The conditional independence between outcomes giving the covariates and random effects. The second assumption is commonly used to handle informative censoring, in which the censoring is allowed to be conditional with correlated with the outcome process and the self-reported process through the random effects. However, after condition on the random effects and the covariates, the censoring is independent of the barrage processes. So the fundamental step of the pairwise likelihood in our previous work is to consider two observations from two different subjects. However, the two observations could not be comparable due to the informative censoring now. So we cannot use the previous pairwise likelihood anymore. So to bypass the challenge due to the incomparable pairs, we consider two observations from the same subject. See the J's and K's observations from the same individual I. Then the conditional density of such a pair, giving their order statistic, only depends on the observed data and the parameters of interest. So the conditional density does not involve the random effects. Thus, we do not need a parametric distribution of the unobserved random effects. So this equation represents the contribution of one pair of observations. We can consider all possible pairs of observations from the same individual by taking the product of all possible conditional density while leaving out their correlations. We obtain a composite likelihood function termed as revised pairwise likelihood. So you may notice that the major limitation of this proposed approach is that the effects of baseline, the time invariant covariates, are not estimable by using this likelihood, since they are limited by the above conditional procedure as well. So the method is only applicable to studies in which the research interest is to evaluate the effects of time invariant covariates. For example, in the weight loss program, the covariance of interest is the usage of the program, which can be categorized by number of logins or number of blocks up to time t. Currently, we cannot figure out a ideal solution to this limitation and how to live with this, for our future research. So if you have suggestions, ideas to share, more than happy to discuss with you further. So now let's look at some simulation results briefly. We compared the performance of estimators from 4 methods: the GE method, joint modeling of longitudinal outcomes and informative censoring, the pairwise likelihood in our early paper, and the revised pairwise likelihood. Here, I only present the results from 2 scenarios. Under the first scenario, the observation process is independent of the longitudinal outcomes, while under the second scenario, the two are dependent with each other. Under both scenarios, the censoring time are correlated with outcome, with longitudinal outcomes. So we can see the revised pairwise likelihood provided desired estimation accuracy and reasonable coverage probabilities under both scenarios. Since the GE and the pairwise likelihood cannot deal with informative censoring, so both worked poorly under the 2 settings. The joint model implemented in the R package can properly handle the informative censoring, but cannot deal with the outcome-dependent sampling. So under the first setting, the joint model works well as expected. Also, its standard errors were smaller than those of the proposed method, since all distribution information was used in the joint model method. However, the efficiency loss of the proposed method was around 15% only, was not that much considering the robustness of the proposed method. So we can see the joint model did not work under the second setting. We have applied the proposed method to the self-reported weight data. So the number of logins, the number of weight entries ranged from 2 to 168 within 1 year. So we applied 3 methods and consider 3 time varying covariates. The number of login to the program up to time t, which was used as time varying measure of the usage of the program. Length in the program since the enrollment up to time t and its square term. So we found that all 3 time varying covariates had significant effects on weight. So usage of the program was positively associated with weight loss. We also found that participation length has a positive effect on weight loss as well, although there is a long-term rebound effect suggested by the positive sign of the scrotum. The estimated program effect by the revised pairwise likelihood was smaller in magnitude than those by the other two methods. This is not surprising, ignoring dependent censoring or by sampling in this application tended to overestimate the effect of the program.
Jing, sorry, quick question about the number of logins variable. Is it a cumulative? So up until that time of that weight loss measurement, the cumulative number of logins to that point?
Right, exactly right. It's a cumulative number of logins up to time after the weight entry. Yeah, at time. Yeah, okay. Yes. So let's move on to my last example about bias sampling. The publication bias in meta-analysis. So meta-analysis is a technique for combining aggregate results from multiple independent studies. By combining information, meta-analysis usually has higher statistical power than a single study. However, the validity of the results of meta-analysis is threatened by a publication bias. So publication bias occurs when the decision to publish the research depends on statistical significance of the results and other factors, such as sample sizes of studies. A great deal of the efforts have been devoted to developing statistical methods to detect and correct for publication bias. One popular method is a selection model that imposes parametric distributions to categorize the underlying publication mechanism. For example, the corpus selection model assumes that the probability of publication depends on both the effect size and its standard error. So specifically, the corpus model includes two components. One is a model for the estimated effect size, and the other is for the publication process. In the first model, the theta hat and the theta are the estimated and the underlying effect sizes. The parameter tau describes the between study heterogeneity. Then a separate selection model uses a latent variable Zi for the publication process. Here, the study I is published if the latent variable is positive. Different from the complexity of self-reported bias in the second example, the publication bias is generally believed to depend on the estimated effect size and its precision. So in this corpus model, the parameter gamma 1 characterizes how the chance of publication depends on precision of the estimated effect. Then the correlation parameter rho between two random error terms describes the impact of the estimated effect size on the publication process. So even for this popular model, there are some remaining computational issues. As acknowledged in the literature and confirmed in our simulation studies, directly maximizing the likelihood is computationally unstable because the likelihood takes its maximum over the relative flat surface and often has non-convergence results. Accordingly, the existing R packages use a graded search algorithm for a subset of parameters. However, the up package ignores such additional variation due to the great search algorithm and provides underestimated standard errors. Which could result in potentially misleading inference. So to tackle the computational challenge, we propose an EM algorithm to obtain the NLE, which recasts the bias sampling issue as a missing data problem. So in our proposed EM algorithm, we treated the unpublished studies as missing data. So then the variance estimation is straightforward by using the Lewis method. As demonstrated in the numerical study, the proposed EM algorithm yields stable estimators and reasonable coverage probabilities. So multivariate meta-analysis is also popular in evaluating diagnostic tests, where the performance is often described by a pair of measures such as sensitivity and specificity. So the methods that account for publication bias in the multivariate meta-analysis are very limited in the literature. So we extended the corpus selection model from the univariate setting to the bivariate setting, and then developed a EM algorithm accordingly. Recently, we have also generalized the corpus model and the EM algorithm to the network meta-analysis setting. So, the main messages of my talk is that we cannot automatically apply standard methods when analyzing observed data. So always think very carefully about whether the observed data are representative of the target population. So data with sampling bias are not rare, and the sampling bias is far too common in research. One important area in our research is the dissemination of statistical methods. Recently, we have developed a package for analyzing length bias data, a special case of left truncated data. So this package includes statistical methods for testing the length bias assumption, model fitting, and model diagnosis. We will continue our efforts in this direction for other types of biased data. So that's all. Thank you.
Thank you, Jing. I'm going to ask, does anyone have any questions? Please use our Q&A function. So, sorry, I didn't know, you mentioned that other biosampling cases such as nested case control, are there specific like data, because I noticed a lot of your projects are motivated by actual data sources where there's problems in the data and you're trying to fix them. So for the nested case control cohorts, do you have specific examples that you're worried about where these biosampling occurs?
Yes, so when funded cancer early detection project called as THCCC actually uses the nested case control design for connecting biomarker, high-dimensional biomarker information. So, actually, in current statistical literature, some methods have been proposed to use adjust the outcome-dependent sampling bias due to the nested case control sampling. However, with the high-dimensional biomarker data in the discovery phase, we do not have existing method to detect predictive biomarkers, giving the existing biomarkers or existing risk factors with consideration of the biosampling issue. We are currently working on this area.
Okay.
Yeah, to adjust the sampling bias in biomarker discovery phase.
Yeah, yeah. So if there are no remaining questions from the audience, then we'll thank Jing again. Virtual applause.
Thank you.
And yes, it was very interesting, very informative, and I think we all learned a lot about thinking when we look at our data and where it's coming from and what are the biases we must be careful about. Okay. Thank you very much. Thank you, everyone.
Thank you.
Thank you.